Mhm. So again, let's continue. I'd like to start with nitrogen. What's this deal? It's actually a very big deal. It's necessary for all of life. Everything living consists of protein structures, and our amino acids are the building blocks of those structures. So as you can see here, nitrogen contributes to the structural backbone of all amino acids, and they can also contribute to the R groups of select amino acids as well. It's even a part of our DNA. These are the four nitrogenous pairs that make up our DNA code, and they're considered nitrogenous bases because nitrogen is a key element in forming their cyclical structure. Nitrogen is also a limiting factor for plant development. You can have all the other essential chemicals present for the plant to use. However, if you're lacking in nitrogen, you won't be able to have your plant develop as well as you'd like. So that'll bring us to root nodulation and nitrogen fixation. What are they? Root nodulation is the formation of tumor-like growths on the root system that house nitrogen-fixing bacteria. So in this case, the picture exhibits the root system of a legume plant and rhizobium bacteria. Chemical signaling will occur between the legume root system and the existing soil rhizobia. The roots will uptake the bacteria into their structures, where the bacteria will begin to proliferate, resulting in nodule formation. Now, atmospheric nitrogen is in the inert form of N2, and plants can't use this form of nitrogen for their development. So the atmospheric nitrogen will permeate the soil, where they will be taken up by the root nodules. From there, the rhizobia can convert the inert N2 into ammonia, or NH3. And it's in this form that the plant can use it for its development. This is a wonderful process because essentially these legumes are like hiring the bacteria to make fertilizer for itself. So now, why are these two concepts important? Well, they develop a symbiotic relationship between the roots, plants, and the bacteria. And this relationship is only exclusive to members of the legume family. The plant is able to provide carbohydrates to the rhizobia, which they can use as a source of energy. And in return, the rhizobia produce usable nitrogen for the plant. This means that legumes have access to another nitrogen source that no other plants can tap into. Looking at the picture, you can see how plants inoculated with rhizobia are far more developed and better off than those that aren't. This is because they can capitalize on that extra source of nitrogen that other plants aren't necessarily able to tap into. There are also ecological reasons to the benefits of nitrogen fixation. There's a great demand for nitrogen fertilizers in agricultural use. Artificial nitrogen fertilizers have lasting environmental impacts, and the graph shows in nitrous oxide, which is a greenhouse gas released as a byproduct of fertilizer production. Nitrous oxide has been steadily increasing since 1980 and still continues to do so to this day. However, symbiotic nitrogen fixation is a natural process that provides a more sustainable form of fertilization. So I'd like to introduce to you Antoon Ploeg, who is a professor of CE in nematology at UCR. And one day he was looking for nematode structures on the root system of this plant. He didn't find any nematode structures, but he did find some interesting growths instead. So he dropped off the plant at our lab. And that's how we were able to obtain our first sample of Phytolacca acosandra. So these are the growths that were on the initial sample of the root system, and they do look similar to that of legume nodules, but we're a little bit unsure. So this would be interesting if they are nodules, because Phytolacca acosandra is not a legume. They're actually very distantly related to the legume family. Phytolacca acosandra is in the order Caryophyllales, while legumes are in the order Fabales. So we wanted to test if this distant cousin can engage in symbiotic nodulation. So how do you go about determining if a microbe causes a disease? Well, Robert Kochs came up with a set of guidelines to help answer that question. You could think of the root nodules as the disease and the bacteria as the ones causing the disease. So our objectives will mimic his postulates. So for the first objective, we wanted to see a disease organism that is present with an abundance of a microbe. Objective 2 is that the microbe could be isolated from the infected organism and cultured in media. And objective 3 is to introduce the microbe to a healthy organism, which should cause the disease. And our last objective would be to re-isolate the microbe from the infected test organism. So where we are now. From the initial sample, we crushed up those growths and had their contents streaked on growth plates. After inoculation or an incubation period, growth was observed, and these cultures were identified via blast analysis using the ITS portion of their DNA. Of all the bacteria that were identified, you can see the ones that are highlighted, a significant amount are part of the Rhizobia family. This is great news because Rhizobia are already known to engage in nodulation and fix nitrogen. So on the plant side of things, you need to have test subjects. So let's just say that growing these plants up from seed to get test subjects were essentially a pain. Like, these seeds were 2 to 3 millimeters in length, super tiny, hard seed coat, and the embryo is actually pressed up against the seed coat. So to remove it, I was using a scalpel, but if I wasn't careful, I could damage the embryo, which would result in the plant not growing correctly. And it just felt like I was doing microsurgery on these seeds. And after hundreds and hundreds of processing seeds that didn't take, we decided to go through a different route to obtain our sample size. And so that's when we decided to go down to San Diego, where the plant was in abundance. We took 100 cuttings from existing plants and processed them in the lab by adding IBA rooting hormone to induce root growth from these cuttings. We kept the cuttings in moist soil and covered them with plastic bags to reduce water loss through transpiration, and allowed them to grow. And after 3 weeks, we finally got some roots. The roots are here. Took the better part of a year, but we have roots. So super excited about that. So based off of that information, objectives 1 and 2 complete. Now that we have roots, we can begin inoculation trials. 3 groups would be tested: a control group, a colony group, and an individual group. The control group will be inoculated with bacterial cultures that have been killed off. The colony group will be inoculated with all the bacteria at once, and individual group will be tested, inoculated with individual bacteria. So hopefully objectives 3 and 4 will be completed very soon. Now, what does this research mean? Well, if Phytolacca icosandra can engage in symbiotic root nodulation, then future research can be conducted into the mechanism of this symbiosis. Since Phytolacca icosandra is distantly related to legumes, we can also gain insight into how to incorporate this mechanism to other plants that are distantly related to legumes, such as cereal crops. Those consist of corn, wheat, and rice. By doing so, this would allow us to move towards a more sustainable form of agriculture, ultimately benefiting all of humanity. So we have the corn, wheat, and rice. I'd like to take this time to thank my team, Dr. Jewel Sachs, who's my PI, postdoctoral researcher David Frank, and Arfat Rahman, who just got his PhD too, so congratulations. All of which I've learned so much from, and I wouldn't have been at where I am now without them. So thank you, everyone. That concludes my presentation.
Thank you, Anthony. Don't forget to stop recording.